this session, the first part of this session is going to be what's new in CCP-EM, EBIC, and the EM databases. And we have one person to represent each of those who are going to give talks. So these people have really been doing enormously helpful, valuable jobs for the whole community, and they're each going to give us an update. So the first one is Agnel Joseph from CCP-EM. Just to give a brief background, he has a PhD in structural bioinformatics, has done computational methods in cryo-EM, did a postdoc at Birkbeck, and he's now at STFC working on EM validation for the CCP-EM project. So Agnel, go ahead.
Yeah, thank you, Helen. Just share my screen. Yep. Can you see my slides?
Yes.
Yes. Okay, great. Yeah, thank you. So I'm going to talk about CCP-EM. So CCP-EM stands for Collaborative Computational Project for EM, electron microscopy. And like other CCP projects, CCP-EM also aims to develop a UK EM community and help and support software users and developers. So we work with a network of software developers and try to integrate their tools in a user-friendly interface. We are based at STFC, and we are located close to Diamond, EBIC, and RFI. We share an office with CCP4, so it's quite convenient. So we share expertise with crystallographers in CCP4. So at the moment, we have two core staffs, Tom and Colin, and I work on a validation project. Yola is the machine learning expert in our group, and Martin Wynn is the group leader. So just to give an outline of the software suite that we have developed. So the software suite is meant to integrate tools for different EM data processing. And it's built on a Python framework. And you can get the software for Linux and Mac from the CCP-EM website. So the latest stable release is version 1.4. But for any recent updates or recent developments, please download the nightly builds. It is free for academic use, but industries are charged for downloading and using the software. And any bug reports or feedbacks, please email ccpm@stfc.ac.uk. So an ideal workflow going from images, raw images to final atomic models would look like this if you were to use CCP-EM tools. So you start with Relion for image processing and reconstruction, and go on to map optimization and model building and refinement and so on. So I'll take you through different stages of this workflow. So we have Relion packaged in CCP-EM distribution. So the version 3.0 is in CCP-EM 1.4, and version 3.1 is in the latest nightly build. And at the moment, the Relion stands a bit separate from the rest of CCP-EM software, but work is ongoing to integrate Relion more closely with CCP-EM. So you will have a common pipeline and an interface in the future. And we have Relion 8 script, which is kind of a wrapper script to schedule jobs for automated on-the-fly processing. So you would have heard about this from Yun yesterday. And this is being used at EBIC at the moment. And Colin from CCP-EM worked for developing this along with members of Shaw's group and some of the EBIC staff. So once we have a 3D map reconstructed with Relion, we can go on to do further map processing and optimization. So there are different tools in CCP-EM which will help you with that. So a few are listed here. So the MRC to MTZ is utility which is part of RefMac and developed by Garib and co. And it basically helps you to sharpen or blur your maps with a range of sharpening factors. So you can basically play with different map sharpening and blurring and see what happens to your map, both in the amplitude spectra and in real space. And the three other methods, laughter, confidence map, and log scale, look more at the local details of the map and try to denoise or filter the maps. So you will hear about laughter from Chris on Thursday. Laughter is basically a method for local map filtering or denoising. So if you give two half maps and a mask where the calculation will be done, laughter will give you a more clean or denoised map as an output. So the map from laughter would be more interpretable, and it should ideally aid in model building and so on. The confidence map approach is developed by Max Beckers, and it also looks at the local information of the map. So it tells you, it gives you a confidence estimate of what is the chances of having a positive signal per pixel, per voxel, with respect to the background noise. So it's quite useful to, for example, decide on a threshold for the map, or derive a mask for the map, and so on. Log scale is a local map sharpening method, which looks at local windows of the map and optimizes the sharpening factors based on the local information of the map. This is developed by Aryan Jacobi. And so once you have cleaner or filtered or denoised maps, you can go on to build models to interpret the information in the maps. So if you have structural information of subunits or components of the map, you can do what is called rigid body fitting. So this is basically 6-dimensional searches of the component structures, the atomic models, and try to fit them and localize them in the map. So you can use two methods, two tools in CCPM for this. One is doc-EM and another is molrep. Both have a similar purpose, but molrep ideally works at high resolutions, and it is faster than doc-EM. So it will basically give you the location and confirmation of a component structure in a 3D map. If you don't have any known template structures, what we can do is to build models ab initio. So if you give, if you have a map and a sequence which you want to build, you can use tools such as Buccaneer or Nautilus to build sequence of amino acids or nucleic acids. So Buccaneer is quite an established method in crystallography for model building. And you can use it for EM as well through the CCP-EM interface. Now, if you fit models in maps, the fitted models might represent confirmation which can be different from what the target map confirmation is. So you would ideally need to adjust the structure to fit into the map. So this process is called flexible fitting and refinement. So for flexible fitting, you can use FlexEM, which is based on molecular dynamics, which is biased to the map density information. So a model undergoes MD simulation and it tries to fit into the map as the simulation goes on. And you can use what is called rigid body restraints, which is essentially restraining components of the models. The components can be individual domains or secondary structures and so on, and try to fit these individual segments of the structure into the map. And FlexEM is more established for use more at intermediate and low resolutions. And the other method is RefMac. RefMac is used for mainly for high resolution model refinement. So it optimizes the geometry and the fit to map information. And it is again an established method for in crystallography. And this is developed by Garib's group at LMB. And you can again use different restraints with RefMac, which can be derived from the template or homology-based restraints. So you can use some what is called ProSmart or LibG restraints to derive homology-based distance restraints for proteins or nucleic acids. So once you have the refined models built in the 3D maps, you can visualize these models. So COOT is a useful method for map visualization, and you can fix the models and do real space refinement in COOT. So we have the version 0.9 of COOT in CCP-EM, and it has more interesting features for cryo-EM map fitting and refinement. And it works in both Mac and Linux. And as I said, it has new features for EM, but it can be less stable because it is not the last stable release. And it is also available from CCP4 version 7.1. So once you have the models built and refined, we would ideally want to validate the atomic model. So we have a interface for doing atomic model validation in CCP-EM. So there are a list of different tools which you can use for atomic model validation. So mole probability is a popular method which basically evaluates the stereochemical quality of your structure. CABLAM looks more at the backbone, basically the C alpha information and the peptide and carbonyl geometry. You can calculate the fit of the model locally per residue with the tool called Tempe SMOC scoring. We can do something calculate fit to data globally. So we can calculate model map FSCs or calculate an average score from the model map FSC, which is called the FSC average. And we can use RefMap for this. And we also have secondary structure prediction from sequence. So we can kind of assess if the secondary structures predicted from sequence matches with what you have built in the density map. And the interface for this looks like this, and you have the selection of methods which you want to use for validation. And the validation interface gives different outputs. So you have global results, which are essentially single numbers for different estimates. So you have the mole probability scores, CABLAM scores, the model map FSC curves, and the FSC average estimates as well. So you can compare fits of different models, for example, and get a comparison of these scores using this interface. You also get estimates of local quality and local fit to map. So here the table gives a list of outliers, which are essentially errors, potential errors in your models. And these are clustered spatially. So you mainly look at large clusters of these errors in your models, and you can try to fix them in COOT. So when you click this fix model button here, it should take you to COOT interface with the list of outliers there, and you can fix the outliers one by one. So I listed many different tools in CCP-EM and how it fits to the workflow, but there are quite a few other tools which you can use for different purposes. I've listed some of the recent additions here. So you have cryo-EF, side splitter, Harrispex, and the difference map calculation, and EMDA, which is again a Python library for local correlation and difference calculation. So there are other tools which would be interesting for you as well. So you can see the list of tools in CCP-EM on the main CCP-EM interface. And if you also look at the documentation on the updates from CCP-EM, you will get a list of different tools updated in each release. So I thought I will finish with an example from the COVID-19 associated case. So here is an example from the SARS-2 virus, and it is the spike protein, which is on the surface of the virus. And spike protein attaches to the host cell receptor, and it's an important target for vaccine and antibody development. So the last couple of weeks, I was basically looking through the different data on different maps deposited in EMDB from the SARS-2 virus. and try to use different tools in CCPM to see if we can get any improved models, for example. So the example I'm showing here is EMD 21457. It's a 3.2 angstrom resolution map of the spike trimer. And it's a pre-fusion form, so it's not bound to the receptor. And one of the protomer is kind of opened up to be able to bind the receptor. So we have the map and the model deposited in the EMDB and PDB database. And if you look at the map is globally sharpened. So as I mentioned before, a globally sharpened map may not be optimal for local areas of the map. It can end up in over sharpening or kind of over blurring areas of the map, depending on the local resolution. So if you look at if you can see my mouse cursor, so if you look at the first row in the figure, it is showing the receptor binding domain of the spike protein. And if you look at the map deposited, which is in gray, it has some broken density areas for this domain. And this is potentially a result of over sharpening for this part of the map. It is locally a low resolution area of the map, and it's over sharpened for this resolution information. And if you there are crystal structures of this domain bound to the receptor, and if you fit the crystal structure of the domain into the map, it fits relatively well here. And if you use a local sharpening method as to a global sharpening approach, you begin to see more features of the map. So if you see here, the map begins to reveal more features if you do a local sharpening. So the yellow colored map is the locally sharpened map.
Agnel, I think you better start winding up now, okay?
Okay, yeah, so basically the idea is that, so you can use the local map processing approaches and information from related structures to build more residues in the map. So I could build approximately 150 more residues onto the model which is deposited. And the reprocessed model has a reasonable geometry and a slightly better fit to data based on the model map FSC. So I would like to stop here with acknowledgements to all the collaborators and developers that we work with. I would have missed some names here. I do apologize, but and if you have any issues, please contact us on these contact details. And thank you for your attention.
Okay, thank you, Agnel. We've got a few questions accumulated for you.
Okay.
First of all, there's a general thing that comes up. People are asking whether stuff's going to be available afterwards, and these lectures are being recorded. They'll be on YouTube, and I think if people want the slides, are you able to provide those?
Yes, yes, I think we will provide the slides for.
Okay, so this information will all be available.
Yes.
So there's a few other questions now which I'll go through.
Yeah.
Julien Bergeron has asked, CryoSpark already implements variations of the map optimization software. Is it possible to jump straight into the docking model building?
Yes, so most of the tasks that I explained here are kind of independent, so you can straight away jump into different stages of this process. Yeah.
Okay. Actually, there's another question from Sabo. Is there a tutorial available? I don't know if you have that on your website.
So we have tutorials from different workshops and courses that we have available on the website. So if you go to the CCPM website, we have tutorials from different model building and Rely-On workshops.
Okay, now I guess there's quite a few more, but I'm afraid I won't be able to do all of them.
Yeah.
So from Piotr Kolata, how do log scale results compare to varying B factor during manual model building in Qt?
So if you look at the B factor distribution, you have essentially high B factors for the flexible or the low resolution areas. And log scale will give you a more blurred map for this high B factor or more flexible areas. So you will see a more blurred density for this.
Okay, well, loads more questions have appeared, but I'm afraid we're running out of time. I think
Okay, yeah, no worries.
I'll just
Yeah,
randomly
yeah.
choose one more from Fabrizio Martino. Is the database of MolRep integrated in the CCPM suite?
Not at the moment, but it will be in the future.
Right. Okay, well, I'm sorry to all the others. Please do keep putting in your questions for the other talks, but we've run out of time for this one. So thank you very much, Agnel.
Yeah, thank you.
And,
Thanks.